So another huge problem with studying something from a genetic aspect and figuring out what causes what is something called epigenetics. Epi, a rough translation of epi is above, so it's kind of above genetics, but what it is it's how genes are turned on or off, or how much they're read in the genome, or how they're read, instead of the actual genes and genetic code being changed oftentimes. And it's the reason why every cell in your body is different. It's the reason why a nerve cell is not a skin cell. It has to do with how you develop, and so on and so on. And it's kind of in between. I'll get to it. It's in between genetics and environment. Your genetics affect your epigenome because it determines on how genes can be turned on and off, what they are. It determines the switches and stuff. It determines how sensitive you are to certain things, your genetics. But your environment can turn it on, can also affect your epigenome by affecting all these things that turn genes on and off. And it can be inherited partially, or it can be rewritten later on in life. And the younger you are, the easier for it to be written, but most easy it's when it's already still in the womb. Now, some of it can or cannot be fully rewritten. And I'll get to all that. But I want to show you just a big overview on how complicated this is without actually going into very complicated science and math, just how many different things go into it. Well, the most basic thing about the genome, epigenetics, is that we have to understand how DNA is read. So DNA, there's something called RNA. DNA is turned into something called RNA, which is turned into proteins. So DNA turns into RNA, turns into proteins. Now this DNA is read to be turned into proteins by some other RNAs. If you want to be specific, they're called messenger. They're called tRNAs or translator RNAs, and which make messenger RNAs help with some proteins. These RNAs then are turned into proteins by something called a ribosome, which is actually another RNA that's super big and fancy compared to all the other RNAs. Now, there's a bunch of ways you can change this up for how genes are read and turned into RNA and eventually into proteins. Proteins are more stable, in some ways more stable than RNAs, and they can do a lot more different types of reactions than RNAs. They can handle many more different environments. They're a lot more specific than RNAs in general, and they can also be stronger and more structural, have more better structural properties for making hair and stuff. RNA is used in the body more so because RNA is something that is a lot more easily modifiable than DNA and can be made a lot faster, and doesn't mean it can be then DNA can, with DNA is halfway directly turned to protein. RNA also protects DNA because the DNA is not much more stable, so you don't want something too unstable like RNA to turn into, to be stored in genetic material or mutate too fast. Very few organisms, the only thing that can really survive with genetic material being RNA is viruses. And that's only certain viruses, and those are the fast mutating viruses because they're so simple and flexible in their genome. Another difference between all this stuff, just to give you some more background, is RNA is usually very similar to DNA, except for it has a single, instead of DNA with this double helix, RNA has this one helix. It doesn't have two things bounded together. DNA has two things bound together from both sides, like A, T, C, G. RNA would just have one letter, not two letters paired together. And the T, which is thymine, is replaced with something slightly different, uracil, but that's not very important. Now, there's also RNA has a different backbone than DNA, which is a less stable backbone, but easier to make. So let's start getting to actual epigenetics. How can a gene be turned on and off? Well, in your body, there's actually meters long of genetic material in each cell, but it's wrapped really tightly. And how it's wrapped determines how easily it turns into RNA, which then turns into protein, so much protein a gene makes. If it's wrapped if it's, first of all, if it's twisted less tightly, like it's so here, it's easier for RNA and proteins to get inside of it and read it, and they can read it faster because they can have like twist and turn less, so less friction, and that means they can produce a lot more RNA. So it's more like they turn on more. Another thing is that not only is it twisted in the double helix more or less tightly, but you can have all these helices are then wrapped around proteins called histones mainly. And there's a couple different types of histones. And these histones occur in pairs, usually of 4, but they can occur in more, groups of 4, they can occur more or less. And there's a lot of variation types, but for our cases, histones. And these histones, then DNA can be wrapped around histones. Now, histones can really block off a large section of DNA, so it's not read at all, but they can also leave some sections exposed more than other ones, and they can be wrapped around more or less tightly around the histones, so they can change it there how much it is easy for it to read, and how much also curves around the histones can affect how easy it's read. This is huge because it has something, we've shown this has stuff to do with cancer genetics, because they're wrapped less tightly around in cancers, because that allows them to make more proteins faster. In fast replicating cells, many times it's wrapped less tightly so it can make more stuff fast, but some parts are more rapidly tightened, more rapid, so they can not be as so that it can be more stable. And wrapping it sometimes more tightly in one area can actually make the other areas wrapped around less tightly, or vice versa, because you want to overall do it. So it's like, and that's more complicated mechanism. But you can imagine that I stretch out something along the histones, and I really stretch it, wrap a string really tightly around 2 things. But in the middle section that's not wrapped, it stretches out and makes it really taut so it can be read more easily. So that's just the basic DNA being modified. Now, what else can we do to modify DNA to change how it's read? Well, we can have modifications to what actually is bound to the DNA. One modification we can have is methylation, where we add this group called CH3 to actual parts of the DNA. So it's not normal DNA, it has these modifications that come on and off, and this is bound tightly to the chemical bond. Another one we can have is acetylation. And acetylation is we add this carbon with a double bond of oxygen and a single bond to an OH group. And the H can come off and become O minus. And that O minus the negative charge can go to one oxygen, and the other oxygen can be bound in something called resonance, which can stabilize the structure. But that means acetylation makes it DNA more negative. And it makes it also more acidic. Acetylation comes from acetic. Acetic acid is actually what makes vinegar, funny enough. Methylation is adding a group like methane, and it makes it the DNA less charged. This charged things can, making DNA less and more charged can affect everything. It can make it so proteins and RNA stick to the DNA more or less. RNA and DNA are both negatively charged, so putting acetyl groups can repel RNA and make it bind less strongly to DNA. Methylation can do the opposite, but then sometimes they can physically block out section. If you have too much, a little bit of methylation can turn on a gene, but too much methylation can actually turn off a gene by preventing stuff from actually reaching it just because there's too much stuff there, or it's overcrowded. Or you can methylate nearby regions and make it less charged nearby, which means that stuff comes closer, but that stuff is blocked. But stuff comes closer, stuff that's still charged, and reads it. Or in other ways, it's reorganizing how stuff is read. Now these acetylation and methylation groups also interact with how tightly the DNA is wrapped, because how the charges interact in the whole DNA causes it to twist more or less in different regions, and that's really complicated, and we don't fully understand it, but we know it's real. It also causes to interact differently with the histones, because histones themselves have a positive charge, so now a negative charge can make it interact more or less strongly with the histones and react more or less tightly. And histones themselves can have acetylization and metallization and change how strongly they interact. Your body can modify histones in many ways. They can do chemical modifications. There's different types of histones your body can make. They can make more or less histones. They can change the location of histones. So you have all these different things and all interacting together into some complex ways. But this is just a rough idea that they repel or attract things in a very complex way. You can have RNA. So RNA can bind to DNA, but RNA can get kind of stuck bound to DNA and block large sections of DNA from being read. This is a very big important mechanism and actually in sex determination and sexual dimorphism and stuff like that, and sexual characteristic development in females, where they have, and specifically mammals, other females and other groups do not have this nearly as strongly, but most most human females have XX. One of the X chromosome is almost fully turned off, and it's done a lot by having lots of RNA that just bounds around the DNA, one of the X chromosomes, and just stops anything from being read. And this RNA can also change the whole shape of the DNA and make it wrap around itself more tightly and twist it by putting more strain and tension in the areas. It can also do the opposite, and it can open up areas by twisting it in different ways. Or it can make this so much strain in one area, the other area gets released again. It all depends on exactly how it's bound and how it interacts with other things. And that also then the RNA is also interacting with acetylation and metallization, this bound RNA. So it's a whole complex thing of all these different parts interacting together. Then you can have proteins and other things that are temporarily bounded to these. And these proteins, I already talked about histone, but there's many other ones that are just more local and don't have as big of effect on the global structure, but more on the local specific structure that turns it on or off. These proteins can work by changing the overall shape of the DNA, or they can just attract other things to the DNA, so that they can attract RNA, they can attract proteins that read the DNA, they can do stuff like that, or they can make it DNA more or less flexible. There's even some ideas that they might change how DNA vibrates, because all molecules are safe in your body, and maybe they change how it vibrates, leading to more or less access to certain regions. So a region might vibrate so it's more open, or vibrate too much that it's less, that stuff can't attach to it well because it's always taking it off. These are all, that's not proven, that's just an out there idea. But you have all these different things and they're all interacting together. It's not a simple thing they can really easily take apart. We've learned these mechanisms by looking at oversimplified examples without that much environmental factors. And also from a lot of data for the extreme cases. We do not understand them very well in human beings, except it does happen definitely from skin cells, changing a skin cell to a nerve cells and vice versa. And we have evidence that all of this is present. So that's just what we can do to DNA on one hand. Now, what can we do outside of DNA? Well, your RNA can also be changed up. I'm going to go over a couple different ways RNA can be changed up. One way is that RNA can have something called introns. And remember remember that RNA usually was a single ladder. It wasn't a double helix. It was just like one end of a ladder with some stuff sticking out. Well, those things can fold in on itself. And when they fold in on itself, in some places, and make a knot, that knot can be snipped away. Or the knot can that can make a shorter RNA, because you cut away some of the RNA and change how you read the RNA to make a new different type of protein. So same RNA can make different types of proteins. Or the whole RNA can just disappear. Or that it can make it just hard to read because there's a knot in it, so it interferes with it being read. This is how it's done is really complex. There's also many different ways it's been done. But the human genome has a lot of these sections that can be cut out in once it makes RNA or inserted or changed up. And then we have the interfering RNA mechanism, which I briefly talked about in my video about genetics, the first one. And that one is simply that, remember, it's a single RNA is single helix again? Well, single helix RNA can bind to another single helix RNA, and that can make a double helix RNA, which then the body sees and then destroys. This is also how the body gets rid of some RNA viruses. Some RNA viruses have double RNA strands, and they use it to figure out that it's a virus and destroy it. But your body can use that to get rid of certain RNAs and regulate how much RNA is. And it can be switched on if you have too much RNA to turn it off. Now, one of the two last mechanisms of RNA, how RNA can affect itself, how it's read, is that we can add more or less stuff to the RNA. So the RNA is usually read, and we call this the open reading frame, is usually a part that really makes the protein. But we add stuff before the RNA, a cap, and we add a tail. The longer the tail, usually the more stable it is, but it can be slow to read. The cap also stabilizes it. And different ones are different amounts stable or have different reading speeds. And the more stable RNA can be read more times, or it's less likely to change. Also, sometimes the cap and tail can change how easy it's cut up in different places. And that can always change how much protein each RNA produces, what type, and over what length of period of time. Another thing is that RNA itself, and we're only beginning to understand this, have chemical modifications. Just like DNA, it can have acetylation, methylation, and much, much more. And we don't fully understand that, but it does significantly impact it. Strangely enough, the highest amount of these odd things going on is in cephalopods we know of, and it's like almost all the RNA has weird modifications, and the genome is much weird and complex to a human being, beings and mammals and other creatures, and we really don't understand it. And it's mostly in their brains, and they have some of the smartest creatures, and they have a brain that's not like anything like mammals or any other creature. Totally different way of doing it. They have some of our wiring is some of the most complex, but they have some of the most complex chemistry. And we really don't understand what's going on with their brain. They should not be that smart. So it's probably part of the mystery. So that's what can be done in RNA. Now, one final thing to happen is we have something in the body called jumping genes, which I actually did briefly talk about in a previous video. So what are jumping genes? Well, some genes can replicate themselves and copy and paste themselves somewhere else, or just cut themselves and paste themselves somewhere else and move around the genome. This is actually how your body makes all of its antibodies. We think it's how your body makes stuff for you to have certain smells and be able to smell certain things. There's other things that we know it has something to do with also sex determination. And we have strong evidence more in other organisms, like corn. The colorful corns with all the different colored things occur because of jumping genes. Jumping genes are most active while an organism is just an embryo. And the earlier on in fertilization, the more active they are. And it also might lead to we also know it leads to different skin pigmentation in animals and fur pigmentation, maybe in human beings, with birthmarks. But all of this can be affected directly by the environment to some extent. However, to some extent it is totally erased when, not totally erased in sperm and egg cells. But it isn't totally erased because there's feedback loops. So sometimes a little bit of RNA is left over, but an amplification process of that little bit of RNA comes and magnifies it a whole bunch. Another one is that, or a little bit of protein is left over, because proteins also affect this whole thing and can cause more or less splicing of certain things, and a little bit of proteins left over, and we have some evidence of that in yeast, and that causes the whole thing. Or something is not perfectly erased and it's just amplified or degraded in certain ways. So that can lead to a lot of complications. We also know that you inherit different epigenetic molecules from males and from females that formed you. And that can lead to one gene acting totally differently, depending if you inherit from your male parent or your female parent. And we see this in mice with actually diabetes, and we've seen it with a couple genetic syndromes like Prader-Willis versus Angelman syndrome, which can have very different phenotypes. Which phenotypes is how they physically appear. One usually causes very skinny, people excessively skinny. One causes excess fat. So it is possibly one. It's also, we think it might lead to something called stochastic determination of stuff. And stochastism is a fancy way of saying random stuff. And basically, with all these mechanisms, very early on in childhood, a gene might randomly decide to turn on and off and stay on and off. And then you'll have this whole thing for the rest of your life. And a gene may or may not cause something to appear just because of random chance. It might just randomly be turned on and off. And to get why these gene things might be run is because you have something like so sensitive like flipping a coin. But then imagine that you flip a coin early on and it decides to be heads early on. Totally random chance. But then everything else is just copying that one coin being heads as an amplification product, or tails. And that's how like a very small random thing in the beginning can lead to huge developmental change. We know this is somewhat true with certain things with how cells develop into other cells, like skin versus nerve cells. But we don't know if in the rest of the body. I think it might be the missing piece of some complex genetic traits and how they're inherited, and why we can't find the gene is that stochastic things in the inheritance process are messing up and turning on and off certain traits very early on. But how does this all affect studying? How does all this affect studying autism or something with genetics? Well, it means that the data is even not as good because the gene might randomly cause or not cause autism in different people. Also, the genes also affect how epigenetics works with you. They can make your body more or less sensitive to other epigenetic changes, or more or less in different scenarios. So switches and the amplification process can change. This adds another layer of complexity to studying it, which means the data is not even as good. You're more likely to get false positive instead of real positive and understanding the system. And this whole thing is so complicated, and we don't understand it. It makes it even harder because it's not like a complicated system we understand. Well, thank you very much for listening. Please like and subscribe. I'm trying to reach 1,000 subscribers in a year and 4,000 watch hours, which is really hard, but with your help, I think I can do it. Also, if any ideas for future videos, comments, or suggestions, please leave in the comment section below. Again, thank you very much. Goodbye.